So I'm probably going to regret this. I'm going to try to present two projects, right? But I won't, I may not go into as much mathematical detail, just to give you the sort of ideas across. These are two things that I've been interested in, that I've been working on recently. And actually, so the first one is about cancer. And I wanted to add the second talk, which is peripherally related, but it has more of the stochastics, right? The first problem, the first cancer project I wanted to talk about actually doesn't even have stochastics, although the path forward of adding stochastics should be very clear, right? So, but I wanted to present this. So I'm just going to tell you the sort of outline. This is sort of the spoiler of the talk, so that when you go through it, when I go through the slides, you can sort of identify, right, the answering of these questions, or these statements, really, like where does it come from? So identified. Okay, cancers are, so I want to talk about cancer mutation is the first project, and the second project is related. It's about cell populations and birth-death processes. So the first problem is that cancers are often, and this, even though it doesn't have stochastics, it is hopefully a rare event, that cancers are identified by sort of multiple specific mutations now, right? And these are all mapped out in a lot of mutation in a lot of different cancers. I want to give an example of a cancer, myeloproliferative neoplasms, where the cancer is diagnosed by having two mutations, a JAK and a TET mutation. And it turns out that the order of acquiring the mutations matters a lot in terms of the prognosis, but also in terms of the gene expression level of the cells. Now, all these cells have both mutations, but the lineage in which they acquired those mutations, the past history in which those mutations acquired, changes how their gene expression levels. And then our idea is to well, it's kind of not surprising. So you need some kind of memory, right? If the cell's lineage, mutational history matters, right? There's some kind of memory. And in order to get that, a typical gene expression model with multi-stability can explain these phenomena that have been observed clinically. And that's what I want to present to you as an idea. It's a very rough mathematical sketch. And then the second project was related to birth-death processes. And I want to talk about regulation. And this has been motivated partially by the cancer project, as you'll see, but also by T-cell regulation that I've been working on recently. Okay, so let me get started with the multi-hit models of cancer. This goes back from starting, I think, the 50s, 60s, 70s with Knudsen, Armitage, and Dahl. These are statisticians who came up with models of cancer, right, which are based on successive mutations. So the classic way that this was described, this is very crude, this is from many 50 plus years ago, is just to assume some Markov model for waiting time, or some waiting time distribution of the ith mutation since the last, right? The rate of acquiring mutations, rare mutations. And let's say once you acquire L+1 mutations, then things fall apart and you've got some cancer. So the total waiting time distribution, assuming all the rates are the same, right? This is obviously not true. It's just this function, right? The sort of distribution here. And then we can calculate the mean. And then this is really related to age of onset. If you just look at different cancers, you look at age of onset. This is like a very crude, rough, fitting, but it's really just age of onset, really not Kaplan-Meier technically, but the age of onset of certain cancers, and that sort of provides an estimate of how many mutations this cancer, right? You can imagine it's very crude. And a lot of them are 2 to 10, right? That's using this kind of sort of back of the envelope estimate. And Chou is typically the common one. But this kind of model, if we look back at it, it has no reference to the cell population. It is just your body, right? An individual, how many cancers are acquired. So the more modern aspect of all this is to look at gene expression of these different cells, right? So there's no reference to the populations of cells with different mutational states. The mutations are assumed sequential. It does not distinguish mutation types, right? Except maybe if you had the mutation rates different, but they were typically assumed the same for simplicity. The other, the order, right? And this is what I want to get to. Order of specific mutations are not considered. And so the way mutations are acquired, right? We assume there's an individual and it looks like just the source, right? Just the rate of a mutation coming in. But mutations in cells actually occur through cell division, right? Through the division process. So it's only a daughter that can acquire a cell. This is why I mentioned mutational lineage, right? Rather than saying a person will have n number of mutations existing in them, but a cell will have one, and then the daughter will acquire the other one, right? So it's really the lineage that matters, and we need to include that kind of thought process. We need to include that process into the model. So here's the example. Myoproliferative neoplasm, two types of mutations, this JAK 617 is a common one, actually, and then a TET, right? So around 10 years ago, there's sort of a start of some clinical observations of data collected on patients. And they were described as TET first or JAK2 first. So you take patients, follow them, take samples. Of course, there's sampling uncertainty. If they just have TET2 cells, TET2 mutated cells, and then later on they have both TET2 and JAK2, then you call them the TET2 first patients, and vice versa for JAK2. And then the ambiguous patients were not considered. Okay, the ones where you sample them and they had both, let's say. Okay, so here are the clinical differences. In the samples, the populations of cells, all right, the TJ and JT are cells with both mutations, the JAK and the TET. So the relative numbers are different, right? And you can acquire additional mutations, which I'm not going to consider here, right? So basically, the proliferation rates are different depending on the mutational history. The prognosis, the survival. So TET2 first is up here. This thing is working. The top curve of the Kaplan-Meier curve, right? The top survival is much higher for TET2 first patients than JAK2 patients. So those are gross clinical observations. And you can say, well, you know, the patient has cells with both mutations. Why does the order matter, right? So let's define gene expression levels. That was also another difference. And I'm going to define X as gene expression level, let's say rate for gene X, capital X, right? Which is X naught. And so X star is sort of steady state. Let's assume it's steady state. So O means no mutations. J is the JAK cells with just the JAK2 mutation. T is the cells with just the TET. And then JT or TJ are cells with both mutations, but they came from lineages. They came from a mother that had a JAK mutation only, or a mother that had a TET mutation only and acquired a JAK. Some more details on this. There's a series of papers by this Ortman group out of Harvard. Okay, so the key observations, let me just go through these. These are, I mean, the first one is the order of mutations, right? Non-commutivity. So JT, so they've measured gene expression levels. X star of JT is different from X star of TJ. That's the first non-commutative. Then you can imagine all the different genes that they measured, all the sort of Boolean combinations, right? Some genes are upregulated or downregulated by Jack only if the TET is or is not there, right? So that would correspond to these kind of levels here for the gene expression levels for gene X. They measured some other genes where the TET serves as a controller, a switch, right? So they're upregulated by Jack if TET is absent, but downregulated if TET is also present. And then I mentioned 4 and 5. I mentioned 4. 5 is similar to the Kaplan-Meier curves. The age of JAK2 first patients are younger, right? At the time in which they acquire both mutations. So the time of acquiring both mutations is younger for JAK2 first patients. So these are gross differences. And the idea is that, like, how do we explain this non-commutivity? Basically, observations 1 and 5 in the previous slide. Non-additivity for the cell mutations 2 to 4 in the previous slide at the cell level, right? So here's just a schematic. These are numbers of cells, let's say, right? We're moving away from the individual to the cell. We're looking at no mutations. Then some daughters acquire either a J or a T, and then they acquire the other one. And then it looks like the cells are the same here, right? So it looks like you could say that's the same state, but in reality, they're different states. So now you think, you just look at this picture, it's taking two paths and it's ending up with the same state, or it's taking two paths and being different. So what kind of structure do you need? Have that? Well, you need some kind of memory or hysteresis, right? So this naturally arises through sort of a bistable model. So let me just roughly go through this. So for gene expression, you can typically write something like this, right? Something self-regulated, just at the sort of lowest level. And in 1D, I can write it as some kind of a gradient of a potential. One-dimensional, a very simple system. So F is some kind of self-regulation term. This is typically a Hill function, right? You have the right-hand side as a Hill function, and then minus X for degradation, right? And everything is 1 here, so no units. So instead of a Hill function, I'll put in this function down here that has the same shape features, just so I can make it simple plots, right? Instead of I put a Hill function, I've got 2 or 3 other parameters that I need to present in this talk and make it more cumbersome. So we put in this function that sort of is a proxy for the typical Hill function that you're maybe more familiar with. So in the 1D system, I can have this potential, right? And the lambda here is a basal production rate of that gene. And I'm going to assume that, and this is a fairly reasonable assumption, that is what's affected by mutations. Okay. So if I'm doing it, so now the lambdas, right? Lambda equals 1, 2, or 3, it tilts that potential, right? So it makes you want to be the gene expression, right? The minimum is either at 3 something here or closer to 1, right? And so on the parameter plot here on the right, you see that this is the production rate, and this is the 2 branches of stability. So I'm going to say that the green is, let's say, mutation. Okay, so if I have a green mutation, the green arrow is 1 mutation, and the red arrow sends the basal production rate back in the other direction. So if I have the green first, then I go to the upper branch, and then I acquire the red mutation, then I end up in the upper branch, right? Now, if I start in this sort of bistable region, and then I have the red mutation first, either J or T, and then I impose the green, then a daughter gets the green mutation, I go back to the same branch, right? So this is one of those cases where there's no difference. So, okay, you think, well, this is the gene expression landscape for one cell, right? And I just said that the cells only acquire the mutation when it's from the mother, when it becomes a new daughter, right? So the assumption here is that the landscape stays the same of the daughter, right? The gene expression landscape, except for the lambda term. And the gene expression level, X star, is acquired by the daughter. Okay, the expression level, the amount of mRNA in the cell. So that assumption is basically it's a concentration. The cell gets divided in half. The concentration stays the same. Okay, that's just a simple assumption that doesn't have. I mean, we can relax that, of course, with stochastically. So this shows how non-commutivity can arise, right, in this system. And this is how. So these models I just put in. Okay, there's a basal production rate plus constants with indicators of whether each mutation exists or not in the daughter cell. Okay, so the lambda J and lambda T could be positive or negative, right? So the mutations will increase or decrease the basal production rate of that gene. And if we do that, we can go through the different gene expression levels and propose different functions for this for the production rate, right? And then I switch, using mutations, I switch these on or off. Okay, the coefficients here, the lambdas are just one. And so these forms will give you this non-commutative effect for those genes that show that. The other genes, again, this is just a map of the different forms of lambda that give you the different switching behaviors that I mentioned that were observed, right? Whether the presence of TET2 actually is another valve on the JAK mutation. So all of that can be done. The other genes, there are 10 others that required a slightly more involved model, which is gene X. This is gene X. You have an underlying gene Y that actually feeds into gene X, also as a regulator of gene X. This simple model gives us those other 10 genes that I mentioned that had the TET2 regulating JAK2, right? It said JAK2 will either upregulate or downregulate depending on whether TET2 was present or not. That requires this simple motif, right? So we were able to find these motifs that fit all the experimental data, right? Or at least the experimental, I wouldn't call data, observations, clinical observations. I haven't tried, but I'm a little scared of what the large language models would do if I just fed this information in there, if it would have come up with the same kind of motif. Okay, so then we wanted also to look at the being diagnosed at the younger age, the different cell populations, right? The different relative cell populations. So again, a very simple Moran model could explain that. Okay, as long as you tune sort of the proliferation rates just in a very simple way, right? You assume that certain types, you assume some kind of distribution of fitness effect, really, that after one mutation, you speed up acquiring the second mutation. Okay, and so this was published not too long ago. We're still trying to refine this. So talking about Moran models, that brings me to the second topic that I wanted to talk about, which is so if you don't, I think everyone probably knows, Moran model is just a fixed population. You take an individual and you replace it by its mutant, for example, and you just continue doing this. Moran model is in continuous time. The total population is fixed. So in the Moran model, the birth and death process is concerted. You take an individual, you kill it, and you immediately replace it with the same or a mutated version. Okay, so it has concerted birth and death processes, right? Because the total number is a hard constraint. So we use that just for simplicity. But now let's go back to the birth-death process. I want to understand a little bit the approximations to the Moran model and what we can say about it. So the same starting point is you have a birth-death process written this way with raising and lowering operators for the ith species. Species goes from 1 to D. For the T-cell repertoire problem that I've been working on, the D could be huge. Okay, so you have many, many different clones of T-cell receptors. So in these clonal populations, you can ask questions like, what are the sequence of extinctions? You start with D species. What's the first time to the first one going extinct and the next one? And so this is a coarsening process. It's all happening in all of us after our thymus stops producing T cells. The diversity is decreasing, right, stochastically in these events. So, or you can ask, what is the time to extinction of one of these clones, right, in the field of all the others? And they're interacting. So they're interacting through carrying capacity, right? So I'm going to sort of develop a sort of generalized Lotka-Volterra type model here with a birth rate and a death rate. And you know that you can apply this carrying capacity through a reduction in the birth rate or an increase in the death rate, right? And those cases are described here with a parameter alpha. So this alpha is a partitioning of that regulation I'm putting in. So why do we do this? It turns out in sort of T cell biology, there are many, many signals. They could say increase in apoptosis or triggering, or a T cell gives you a proliferation burst, right? Or there's apoptosis signal. So both things happen. Right? You have regulation that affects both birth and the death. So I wanted to explore, does that matter, right? We know it doesn't matter. This form, it doesn't matter in the mean equations, right? If you write the mean, the dynamics of the expected population, the alphas cancel out. There's this higher correlation there, of course, but the alpha doesn't explicitly appear until the next moment. Okay. Okay. So there are many ways of going about this. I'm just presenting one sort of approach. One is to take this continuum approximation, get the focal plank, right? Write it this way. And the first thing you notice is the drift, I shouldn't call it drift, the advection part is a difference, right? So the alphas cancel out. That's expected. That's what happened with the mean equations. But the noise part, the diffusion part, the alpha's added. Okay. So then you say, well, okay, this alpha, this regulation parameter will affect the diffusion term only, not the drift. Okay, what consequence is that? Okay. So for this particular problem, for the T cell problem, I'm looking at what I will call a near-neutral, or I've defined a new thing called semi-neutral. So with the semi-neutral model, we expect that the birth and death rates, the coefficients, are very close, right? So all the T cells are pretty identical. The T cell clones with different receptors, until they get triggered, they're all fairly identical. So they have similar birth and death rates, but the regulations can be different, right? Certain clones could have different regulations still. So if they have similar birth and death rates, and the birth rate in this case is larger than the death rate, so that I'm pushing up against that regulation, right? I'm pushing up against the carrying capacity. You can construct fast and slow variables. The fast is the total population, and the slow variable is the relative population. Okay. So the total population is just the total size. It's sort of kind of like an Ornstein-Uhlenbeck process right around the carrying capacity. It wants to retract back to the carrying capacity very quickly. This is a diffusive. This is the relative number of one clone in terms of the other clones. That's sort of a soft direction, if you will. So the full Fokker-Planck equation can be written like this, right? With all the terms. This is again large system size expansion, right? Birth rate larger than death rate. The U relaxes, and it relaxes to a quasi steady state total population, right? Which is basically in the ODE case defines the null cline. Right? Okay, 5 minutes. Okay. Okay, so if you do that, you can take the probability, separate them out into just the fast distribution, relax that to U star, use Laplace's method, and just get an equation projected down to the relative population, right? Factor away the U, assume that U is at the null Klein value, U star. And so this becomes the multi-dimensional or multi-species Moran model, right? So the total population is fixed at U star, but the relative populations defined by P can vary. And so the limit of interest is when these values, the birth and death rates, are again within order 1 over k, right? The common carrying capacity value. This is called the near neutral limit, right? And essentially, this is order. These terms are order 1 over k, and there's a 1 over k here. The b is order 1, so this is a Peclet number roughly equal to 1 limit. That's the most interesting case, right? That most people study. But now we have this regulation term that's different. That adds to it, right? That adds to these the standard Moran model. And what you can do now is define an effective population size. Usually, effective population size is used to map the discrete generation Wright-Fisher model to the continuous time Moran model, right? And it's usually like a factor of 2 or log 2 or something. Here, we define a new effective population size that actually depends on the regulation parameter. And you could see that if it's death regulated, alpha equals 0, where all the regulation is in the death term, you get a smaller effective population size than if it's fully birth regulated. And the difference is a factor of B over D, which is a factor of, it's order 1, right? It's a factor of 2 or 5. So this says that, and look at this term, this is the diffusion on the simplex, right? For the higher dimensional Moran model, this projection. Then you have this coefficient is like the diffusivity. So this says that the regulation can control the diffusivity. And what does the diffusivity would control is the extinction times, right? The extinction times of each of the subspecies. And again, it arises because the diffusivity is the sum. It gives you this alpha. This alpha in the near neutral limit gives you this form for the diffusivity or the effective population size, inverse of the diffusivity. Okay. Right. So that's the new result. And we can apply this to look at different extinctions, right? Let's say for the simple D equals 2, that's 2 species birth-death process, or in other words, a one-dimensional Moran process. You have the exact value plus the diffusion approximation. So this is what we're calculating, right? The diffusion approximation to first passage times. There's an error, and the error actually also depends on this effective population size, which in turn depends on the regulation. All right. So this is our result. And this, basically, we got this idea from this very nice paper that I use a lot by During, like Charles During, on the accuracy of extinction times in the Fokker-Planck approximation, right, compared to the discrete. So just a summary, and ongoing work, right? Basically ongoing work, right? So one can consider variation in the gene expression rate acquired by daughter cells, right? So that initial condition that I said, that the daughter cell acquired the X star of the mother, you can put in some variance on that. you can allow changes in that lambda parameter to be stochastic, right? To be, well, stochastic according to the mutation. So it's not just these dominant mutations that are acquired. There's always some variants that are acquired, right? So there's noise around that. So when the daughter acquires a mutation, lambda doesn't change by just a certain amount. There's a probability distribution of what the lambda does, right? So there's some potentially interesting stochastic parameters to play with in light of the bistable models. Also, I only gave the example of a 1D system. Even the 2D system I gave was a potential problem, was a potential flow. But most gene expression models on the right-hand side is not the gradient problem, right? It's not a gradient dynamics. So you have to deal with attractors, and you have to involve with sort of non-equilibrium currents on the right-hand side. So that's also something we want to extend. Again, in this Moran model, there's a number of things to consider, including non-neutrality, stability of subpopulations depending on the different gammas, the gamma ij, the different relative effects of the carrying capacity on each species. Also, what I didn't mention here is we talked about the difference in the exact result versus the diffusion approximation result. And we assumed a quasi-steady state where this was not included, right? So you have these extinctions, these successive extinctions, but the last one takes much, much longer, right? The last one, you have one species left, you need to kill the entire, that's an exponentially long time. But if D is very large, these are very short, and D is very large, the total population of each clone is very small, right? So you can't use the diffusion approximation, right, that I've used here. So you have to go to something more discrete. So there's a lot of rich work to be done, I would say, in this kind of, this type of problem. So anyway, thanks. Yeah.
We have time for one or two questions.
Yeah.
So the theory I'm used to for effective population size, like in the Frank-Fisher model, usually says that effective population size is like the real population size divided by the variation in offspring number. So like if I have more variation, like bigger variation offspring is smaller population size. That's from Gillespie.
Oh, including, you mean having more than 2 daughters?
I mean, if I have individuals that have variation in their number of offspring in like a discrete model, I have individuals who produce variation number of offspring. The effective population size, like inverse to the variation. Is there a way to understand these types of formulas in terms of the variation in the progeny size? If I were to like descriptize time,
right? So the variation. So the way I understand it is through the replacement of the entire generation versus continuous time, right? That sort of samples this variation you're talking about. The regulation effect, this alpha that I defined, I think that's just a separate thing. I don't think it can be interpreted like that. It just came out by the sum. It just came out through the sigma squared, which is the sum of the birth and death terms.
Right, we have one more question here.
With respect to the first part of the talk, what are the prospects limitations for validating that kind of model microscopically? Like, is the limitation more that the biologists don't know what regulates what, or that they maybe know too much and the hard thing is reducing it to a simple model?
Okay. So I wasn't going to look up those genes. I mean, I don't even know if they exist, right? The models for the regulation of the genes, I'm sure they don't, right? There are some models with cancer gene regulation models. I'm still searching for something where I can, So all I could say was just, I'm going to put in a proxy for the Hill function. I don't know what Hill function it is. There are models for that, but they're for different cancers where they haven't really explored the clinical, like the other two hit model is the P53 retinoblastoma pair, those pair of mutations. I don't know that there's as detailed of a experiment or clinical data as this Ortman paper. So yeah, I'm looking. I'm constantly on the lookout for.
Let's thank Tom again.